Okay. Hi, everybody. My name is Febina Mathew. I am the soybean pathologist at North Dakota State University. I work mostly on the biology of the pathogens causing diseases of soybean, as well as developing management strategies to control diseases. And among the strategies, I am heavily focused on fungicides and host resistance. But in the more recent years, I have been dabbling into cultural practices like tillage, residue management, and so on. So today I am going to be sharing my research on Diaporthe diseases. I work on Diaporthe not just infecting soybeans, but I also look at it on other crops that the organism is known to infect, such as sunflowers. We have also looked at it from a wheat perspective, and I have slowly started to venture out into the other crops as well, as far as Diaporthe is concerned. So today we're going to look at Diaporthe Phomopsis diseases. There are several diseases that are caused by the Diaporthe genus. And what I mean by genus is basically a family. We're talking about a group of organisms that can infect soybeans and result in several diseases. So traditionally, or I would want to say historically, these diseases were named based on the plant parts Diaporthe organisms infect soybean. So for instance, if it is infecting seed, then the disease is called seed decay. If it is infecting the stem, it was called either stem canker or stem blight. And as a result of how the diseases were named historically, we know today that there are several diseases. It's not one disease anymore, it's several diseases that Diaporthe can cause. But on the whole, we're talking about a complex, which means a group of organisms, a group of fungi are actually involved in the disease development. Diaporthe diseases have been in the United States since 1940s, and we'll get into that here in a little bit. But as of today, we know it can be endemic in the US as well as other soybean producing countries. But more lately, it has become a big constraint in soybean production, not just in the US, but also in few other soybean producing countries like Brazil. Overall, the impact that the Diaporthe can do on soybean is that it negatively impacts the seed quality and yield. So as of 2021, and based on the statistics from the Crop Protection Network, between 96 and 2021, we have observed Diaporthe diseases to cause a US dollar loss of about 3.63, approximately 4 billion. We don't know if Diaporthe organisms can compromise soybean oil content, but in other crops such as sunflowers, we know that infection of the sunflower plants can typically result in loss of the oil content, and that might be about a 10 to 15% loss. But seed quality is a big, because, and I have a few pictures coming up, but when you think about the and shriveled soybean seed in a plant, or it looks like it's got some discoloration, we know that it's going to be hard for any of the soybean processors to accept those kind of seeds. So in a soybean field, this is what it looks like for a first early symptom. You know, if somebody were to just drive around in a pickup from a distance, it might just look like white mold showing up in random spots. But a closer look would tell us that it's not really white mold. It's a different disease, a disease that has been around since 1920. And the first disease that was reported in the US was spot and stem blight. At the time, because the host was used to name the Diaporthe organisms, it just got one name, and that was called Diaporthe facialorum. And at the time the disease was reported, there was limited effect on crop yield, which means we're talking about 10 to 20% yield loss, and usually observed when the soybeans is kind of ready for harvest. In 1940 came a bigger disease, stem canker, and they found one fungus that was Diaporthe facialorum vorcolivora to cause the disease. It soon spread to the North Central United States, and because we did not have any fungicide recommendations at the time, and farmers heavily dependent on the varieties that were available in the market, they observed about yield losses of 50% or more. And the only way to manage the epidemic in 1940 was to eliminate two of the susceptible varieties, Black Hawk and Hawkeye, which were high yielding at the time, but because they were susceptible to the stem canker fungus, they were just eliminated from the soybean production. So the next few slides, I've got typical symptoms of what these Diaporthe diseases look like. They usually, what we would see would be these reddish-brown discoloration on the stems. There could be some, what I tend to look for is not just the reddish-brown discoloration, but I also look for these black dots that might be present on the plant. And the reason why I look for these is because Diaporthe diseases could mimic several other diseases out there, especially Phytophthora, perhaps white mold, also soybean anthracnose. And so looking for these fruiting structures are very important. Now, for the diseases spot stem blight and stem canker, you may see there are several names out there. And what it means is, once upon a time, it was believed that there was only one pathogen involved. But today, we know that it could either be a complex or one or more of these organisms can co-occur and still compromise the soybean yield. So spot stem blight, as of 22, it's caused by 3 fungi. It could be Lonticola, it could be Sauget, Goulier, and stem canker is caused by 2 fungi, Collivora and Aspergillati. And the most important thing to keep in mind is that it is going to be really difficult to find a soybean plant that's got just one or the other fungus. It's usually a complex. There could be 1 or 2 occurring on a single plant. So this, in this picture, is the reddish discoloration right near the node. That's another place to look for it. And this is really very early symptoms. And as you can see in the picture, the soybeans are sort of entering the R3 stages, past the flowering stage. And that's typically the best time to be looking for Diaporthe diseases. So what I see here on the stem is this reddish brown discoloration that does look like that it's typical of some stem canker. Now, the picture here is more of an advanced stages of Diaporthe diseases. This usually happens when the fungus just decides to infect about 3/4 of the plant. The base of the plant is right here, and then the canker or the discoloration just spreads. This would be in a field where the disease is kind of spread. If you can notice, the soybeans are in their late pod stages, and the disease is sort of spread. It can kill the entire plant with the availability of favorable environmental conditions. Sometimes these Diaporthe diseases are sort of hard to tell from white mold. So what I tend to look for is also spatial pattern of the disease. So as you can see, this particular row of soybean plants are affected. And that's just because the disease spreads like more on a linear basis. It can start randomly in any part of the field, but as the disease progresses, it starts to either be in a linear shape, or sometimes I've seen sort of like crop circles where the Diaporthe disease just tends to spread from all different directions. The picture here is more on the advanced stages of Diaporthe diseases. Sometimes when we see these kind of fields, it's hard to tell what disease would have actually showed up, meaning if it is whether it's spot stem blight or if it's stem canker, it's kind of hard to tell. And if we were to make any isolations in the plant disease diagnostic lab, then we would not get one fungus. We usually would get multiple fungi coming out of the soybean plants. And this is usually a sign when the soybeans would have just gotten ready for harvest. So the one question I usually tend to ask farmers is which of these diseases is Diaporthe. This is very important because as much as in Diaporthe diseases, we notice this reddish brown discoloration, that it could be similar to any of the diseases that is listed here. If you were to look for black dots, you would also find it on an anthracnose affected stem. You would see it in charcoal rot affected stem, or even on a saprophytic fungus. But what is most important is that sometimes when these diseases come together, it's even hard to tell which one might have shown up. So that's why it is important that we send the sample out to a plant disease diagnostic lab that could be a better judge of what disease might have shown up, especially that late into the season. So a lot of the diseases that I have mentioned here, with the exception of Phytophthora that can occur anytime during the growing season, many of the other diseases, bottom stem blight, anthracnose, charcoal rot, or even the saprophytic fungus, all that happens as the soybean enters the advanced stage. So the answer to this question is the first picture right here. That is your Diaporthe disease. Another way to tell Diaporthe disease is when the soybeans have entered the advanced stages and you would have made a dissection on the stem, you would see these black colored inscriptions right at the base of the plant. And this is nothing but just a sign of the fungus, and that is how it appears on the soybean plants. It's one way to tell that it was Diaporthe that caused the disease. Again, this is usually seen when the soybeans are in the advanced stages of their growth. Another symptom to observe, especially when the soybeans are getting ready for harvest, are these white discolorations on the soybean seeds. And that's very typical of Diaporthe. They have a whitish discoloration on the seeds. Sometimes you do see like black dots on them. That would be the fungus basically trying to sporulate and producing some sort of sporulating structures. But this white discoloration is very common, especially when the soybeans are belonging to the later maturity groups, when they have to stay in the ground late before they get ready for harvest. Or it could be that maybe it is on the early maturing varieties, because sometimes when the farmer is getting ready for harvest and spoiler rain shows up, those varieties, if you were to open the pods, you might see some sort of a whitish mycelial growth on the soybean seeds. So the reasons that could really promote these kind of seed infection would be either it's a later maturing variety and there's a lot of moisture, specifically like in the months of September, October, or closer to when the soybean's ready for harvest, or regardless of the maturing variety, whether it's early or late, maybe there is a delay in harvesting them because of a spoiler rain or some other logistic issues. It's important that one opens the pods to see how the seeds are doing. In 2017, it was in 2017 or 2018, there were instances where there was a lot of rain, especially when the soybeans was getting ready for harvest across the soybean production regions. And farmers saw a lot of this white discoloration on the seeds. And on isolations, what they found out was that Diaporthe was probably number one, followed by Cercospora. Cercospora tend to do more of a purplish discoloration on the seed. But white, it could be any other fungus too, but mostly this kind of discoloration is attributed to Diaporthe. So as of 21, what we identified is that there are about 10 species that can cause Diaporthe disease in soybean. The diversity of these species may be smaller in the northern production regions, that is pretty much the pretty much the Midwest, but in the south there seem to be more diverse species composition as far as the Diaporthe pathogens are concerned. So there are, again, about 10 species that are officially reported here in the United States. Now, as much as we went through the symptoms, one of the key things to remember is that it's very hard to differentiate symptoms, identify organisms based on symptoms. So what it means is, like, for example, if I have a symptom of this sort, or if I have a symptom of this sort, it's very hard to look at it and say, okay, that must be Caulivora, or it could be Ascalate. If we were to challenge soybean plants in the greenhouse with an isolate of all the organisms belonging to Diaporthe family, or either one or two, they produce very similar symptoms in the greenhouse, which means either the plant, by looking at the plant, we can tell it's Diaporthe, but it's going to be hard to tell with what exact species may have caused the disease. So ideally, what these fungi are, they are all soil-borne. They can live in the crop residues, and they begin to sporulate typically early in the season. So when the soybeans are getting ready for planting is when these spores are already being ejected from the sporulating structures. It's wind or the rain that usually splashes the spores onto the leaves and stems. And then the lesions the fungi may be on the plant during the early part of the soybean growth, that is between the seedling to all the way up to flowering. However, we don't get to see the symptoms until the reproductive growth stages, which means after flowering. So usually it starts with some level of brownish discoloration on the stem, and then over time, if the rain persists, then what we are seeing is complete discoloration of the plant. And then as the plants are getting ready for harvest, we can also see plant death in severe cases. Now, one of the reasons as to why this disease has become very popular, although the first time it was reported in 1920s to 1940s, and then seed decay was observed or reported for the first time in 1970s. There was epidemic that was happening closer to 2000s. But today in 23, we're still seeing this disease to be prevalent. And the key factor driving them is this climate change. We are seeing differences in temperature and precipitation when you compare what it used to be 30 years ago versus what we have today. Another reason would be the soybean is expanding to new production regions. For instance, here in North Dakota, we may have started off with 1 million, maybe about 25 years ago, but today we are up to 6 to 7 million. So it is really pushing hard to the west. And so as we don't necessarily know when the soil that what was prevalent in those new production regions. Maybe this disease was always there and maybe just didn't pay a lot of attention to it. Third, it could be that we don't necessarily have very solid management recommendations when it comes to managing these diseases. And we're working on it, we're making progress, but it's not there yet where we can provide solid recommendations. So if one were to observe these diseases in their field, there are chances that that we wonder as to how do we manage it. We saw it about 5 years ago. What can I do about it today? As it is again continuing to show up. So these are some of the questions that we have been trying to address through my research program here at NDSU. Again, the species of Diaporthe, they are soil-borne. They can be surviving an infested crop residue, and that could be corn as the previous crop. It could be sunflowers. It could also be on weeds, and I'm going to talk about it here in a little bit. Extended wet weather, usually after flowering, that is from the pod development stage, all the way up to maturity, could be another factor that is actually driving Diaporthe diseases. The infection occurs when the spores are splashed by rain, and a few studies have shown that infection actually starts in the early vegetative growth stages, but we don't see the symptoms until the soybean enters the reproductive growth stages. So as far as the management options are concerned, it's a challenge. We have looked at multiple options and we're continuing to evaluate it. But the key things to keep for us researchers or for farmers or crop consultants to keep in mind is, although we know there are multiple pathogens that can cause soybean diseases, the same pathogens can have multiple hosts. It could be sunflower as a host. It could be the weeds that might be present. There's several of those glyphosate-resistant weeds that are problematic in a soybean field. Think about kochia, think about breadroot pigweed. They're all great hosts of these Diaporthe phomopsis. And on those weeds, we typically don't see Diaporthe phomopsis causing infection in them. They are just going to be living in the weeds as an endophyte, which means we don't see any symptoms on them. Now, I've interchangeably used the word Diaporthe and Phomopsis. A key thing to keep in mind, they're basically the same. Phomopsis is an older version of Diaporthe, and that's the name that most farmers are associated with. But today, based off the studies by Wingfield 2012, One Fungus, One Name, we are calling all the species belonging to this family as Diaporthe. But it's the same. So management options, again, it can be a challenge. It can have multiple hosts. The organisms can have multiple hosts. It's also possible that a single plant, a single host, can be infected by more than one fungi. So what is very important when we think about management options is to be familiar with the pathogens that is likely to build up while soybean is rotated with other crops, because that crop that we may use for rotating soybean can also be a host to these Diaporthe species. Weed management is very critical. There could be dead weeds that can harbor these. There could be live crops. Sometimes we see these volunteer plants, like in a soybean field. Perhaps you have corn as the previous crop, and that just decided to show up in your soybean field. And corn is also a good volunteer for these Diaporthe species. The weeds can also be an alternative host where they can sporulate the fungus and allow the fungus to land on soybean. And weeds can also have multiple species of Diaporthe present on them. I have a few examples here. This is a study that is done in Australia, and we're doing some of the follow-up studies here in my lab as we speak. So in Australia, the production tends to be a little different. They do have soybean and maize, that is corn, which I have highlighted, but they do have others like chickpea, sunflower, mung bean. Chickpeas is very popular in our part of the world, that is in North Dakota. Dry edible beans, field peas, they're all great hosts of Diaporthe. Granted, soybean may not be directly rotated with those, but if you think of the rotation, what we have in the United States is soybean and corn, and their studies have shown where they have isolated Diaporthe species out of corn live plants. So L stands for live, D is dead, S is like surviving plants. And so the one thing to keep in mind is the soybean corn rotation may not help with reducing the inoculum of these pathogens. We don't see symptoms on corn, but the fungus can live in the corn plant and then transmit it to the soybean the next season. And there has been some studies to check what common weeds can harbor Diaporthe phomopsis. And there is a few that has been listed here by the common name as well as the scientific name. And some of the other weeds that is common in our area that also have been identified where Diaporthe phomopsis could live in would be like Kochia lanscota, red root pigweed, wild sunflowers. They're all very good hosts of Diaporthe. Tillage and disking. This is something that we are looking into. This coming year, I'll be having some studies on that. But we are also trying to see if tillage and residue management can help with burying can help manage the disease, manage the organisms. And this is because the crop residues that might lie on the soil surface will allow the fungus to produce structures, what we call overwintering structures, that will enable the fungus to live very long. So if there is any way we could bury the crop residues or we can disk it, we think that could be one way to manage the fungus. So tillage and disking is a possibility. And another thing that we have looked into would be to harvest when the seeds reach 13 to 16% moisture. Once when you dry out the seeds, what might happen is that the fungus does not get the right conditions for it to sporulate and cause the disease. So delayed harvest is one thing. Sometimes the odds are not in our favor because it's raining. I couldn't get my combine out there on time, or the combine broke down. I couldn't harvest in time. And as a result, what happens is the fungus can easily incubate inside the soybeans and cause disease. So it is important that we harvest it at a time when the seeds reach about 13 to about a 15% moisture. Resistance. This has been a big discussion that we are having with the breeding companies. It's very important that we choose varieties with the best resistant package. Typically, the farmers tend to reach out seed dealers, crop consultants to see what are the options that are available to them. Also, the companies, the seed companies. There are few varieties out there that may have resistance to these organisms. So it is very important that one checks to see what the disease rating level is like. But keep in mind that many of these varieties that are available may have resistance only to one organism. So if you're having multiple organisms in your field, or you don't know what organism is actually present in your field, using a variety that has resistance may not solve the problem. And that's just because some of the studies that's coming out of my lab has indicated that the genes conferring resistance to one organism may not confer resistance to the other. So in order to help the breeding companies, and that's primarily because they may not have the parental materials that are having resistance to these organisms, my program, alongside with 17 other collaborators, we decided to look back into some of the varieties that has been previously reported in literature to have resistance to these organisms. And the reason we took this up is one, because we know parental materials may not be available with the breeding programs. Another reason might be because there could be some differences in how these isolates within the same organism can behave. And there has been some studies done in 2003 and then in 2019 out of my program. And currently we don't have much information on the interactions between these resistant genes and the pathogen population. So keeping that in mind, what we did was we went back into the historical literature and we checked to see what has been reported for resistance to these species that we know today. One thing to keep in mind is that if one were to refer to the historical literature, starting from 1920s all the way up to, I want to say 96, 97, the names that has been used for these Diaporthe organisms are completely different from what we have today. And that is simply because DNA sequencing has changed or renamed some of these organisms. So for example, Diaporthe collivora that we know today was called Diaporthe facillorum var collivora. So the historical name does not exist anymore. So as a result of that, there were questions in the minds of researchers as well as the breeding companies. Okay, the parental material that they have reported in the past, does it really hold today? So we decided to revisit the literature to see what parental materials can be recommended to the breeding programs. So as part of this study, this was a greenhouse study that we did. Hutchinson and Century, that was reported to having resistance to Diaporthe collivora. I believe it was reported in like 1980s. This was a PI line. PI 5674473A was out of my research program at South Dakota State, where I was previously located before my current position at NDSU. So were these Tracy M. Crockett Dowling. They were reported by Paoli in 2003 to have resistance to Diaporthe aspergilli. And Longicola is being traditionally known as a seed pathogen, but when upon diagnosis of soybean plants that has been sent to my lab for Diaporthe diagnosis, Longicola is a very common fungus that we obtain from these diseased plants. And we don't necessarily understand what it is really doing, if it's a pathogen or if it's just hanging out there with other Diaporthe species. So regardless, we just decided to screen a few PI lines to see if there's anything that potentially has resistance to Diaporthe longicola. Our susceptible check were Hawkeye or Bragg. And as part of this study, since we were using materials from the past, we decided to challenge it with multiple isolates from different geographical locations for each of the organisms. The inoculations were done in the greenhouse. We inoculated it at the growth stages V2 to V3, which means they're approximately about 2 to 3 weeks old soybean plants in the greenhouse. We used a traditional method in which the toothpick was completely infested with the fungus, and we inoculated it about 50 mm below the first trifoliate node. The control plants were also used, and that didn't have the fungus. And the reason we used control plants without the fungus was to make sure that our plants did not snap because of the wounding that we were doing with the toothpick. The setup was then sealed with the petroleum jelly. Another thing is that when we were trying to standardize the conditions for the greenhouse, we were trying to mimic as much as possible what nature would actually do if we were to screen this under field conditions. So if you notice the temperature for the stem canker fungi, Aspergillati and Collivora, they are somewhere between 27 plus or minus 2, which means it needs a slightly warmer condition than what Longicola would need. So there was a differences in these temperature conditions, as a result of which the experiments were conducted independently. Moisture is a key requirement for disease development. So we misted the soybean plants after inoculations, and then we saturated the pots, meaning we just watered the plants every alternate days. So we used a very simple rating scale. 0 means there is no wound or any discoloration from the wounding. We used 0.5 if we saw lesion spreading, but the plant is still alive. And then we used a rating of 1 if the plant's dead from Diaporthe. So after performing statistical analysis, what my program, again in collaboration with 17 researchers in different states, what we identified were sources of resistance. So Diaporthe aspilari, our recommendation for parental material are Tracy M, Dolling, Crockett, Collivora, PI 5674773B, and Century, and Longicola, it's the PI 417507. So we are going to continue working on host resistance. We do have plans to check to see if there are common genes that could be potentially used as markers that would potentially take care of one or two organisms at the same time. So we are going to continue screening PI lines as well as lines from different breeding programs to identify materials or perhaps elite lines that might be very close to be available for farmers. Fungicides are another option that we typically do recommend against soybean diseases, but unfortunately, we don't have enough efficacy data to suggest that these are specifically working against Diaporthe. So we have some studies happening in collaboration with other states like Iowa, for instance, where we are looking at to see what chemistries work. There has been some work done in my lab to check to see if the isolates that we have belonging to different Diaporthe species are sensitive to the fungicides. So far we have not identified resistance in these populations. So we want to fully believe that fungicides can work, and it may be a question of identifying the right timing, the right rate, and how and when to spray. So those would potentially, in at least in the years to come, we're hoping that there will be fungicides that can work. There has been some studies suggesting that trying to spray fungicides perhaps between mid-vegetative to late vegetative or early reproductive growth stages can help manage Diaporthe diseases, but that's something that we are going to pursue for research in the years to come. But there are specific products available. There might be one or two fungicides that's got pod and stem blight listed as one of the diseases that the fungicide can work against. So it is very important that the crop consultants, the farmers, check the fungicide labels to see if Diaporthe diseases is one of it that the chemical can work against. But so far, we have very limited data, whether it is foliofungicide, seed treatment suggesting they are very effective against Diaporthe. So in summary, we have multiple species of Diaporthe that can cause soybean diseases. So Diaporthe, it's usually a complex. There are several fungi that are involved. One plant can have at least 2 or 3 Diaporthe species. We have isolated up to 4 or 5 Diaporthe species out of soybean seeds, and they could be just spread all over the plant. It could be in your roots, it could be on the stem, it could be on the leaf, seeds, any plant parts. They are just, I want to say, sort of like a merciless fungus. Management options are available, but it's fairly limited. We are researching to identify the most effective strategy that's out there, but currently, if for options, which is important to manage weeds, because many of these weeds, yes, they are glyphosate resistant. The volunteer plants might be again glyphosate resistant, but what is important to know is if we don't manage the weeds, Diaporthe could potentially be a problem in your field if favorable conditions like rain can persist. Tillage disking, that would be one way to bury the crop residues. It not only works for Diaporthe pathogens, but it can also work for other pathogens like Fusarium. So it is important to manage the residues that might be lying on the soil surface. So that's something that we can easily look into before we start planting. Genetics is very limited at this time. There might be very few varieties that's out there that has resistance, but that's something that we are looking into for future. Fungicides are an option. There are several broad spectrum fungicides that typically get sprayed, may not be targeting Diaporthe, but that can help against several other pathogens that are present in there. It may be working against Diaporthe, but that is definitely a data that we are trying to generate. We are basically trying to see if fungicides are effective specifically against Diaporthe. At this time, my lab, we are open to receiving samples from farmers, crop consultants, any of the agricultural professionals out there. We help with accurate disease diagnosis. My lab has developed several molecular assays that will correctly identify the organisms that might be causing soybean diseases. I do have my address there. I also have my email address and my phone number. If there are questions on Diaporthe, I'm always happy to hear and also willing to learn. If there's something new that I have not seen and I need to come out there, I will be more than happy to do that. So for future work, we are looking to see what the fungicide timing might be against the Diaporthe. We'll continue to be looking at different products, new and as well as current products that might be available that will help against the Diaporthe. We're going to continue screening isolates for resistance. At this time, we don't have any resistance issue in Diaporthe that we know of in soybeans. We will also continue to screen varieties for resistance. These might be varieties from the breeding programs that are close to release, or it could be parental materials to help the breeding programs to develop varieties with resistance to the Diaporthe species. As for acknowledgement, I have my lab to thank for and collaborators in 17 US states without which any of the work that I do in Diaporthe is near impossible. I really like to thank the crop protection work for giving me an opportunity to share my research as well as a lot of the photographs that I presented today that come from the crop protection network. I do ask the farmers, crop consultants and ag professionals to refer to a lot of the resources that is on crop protection network for Diaporthe diseases. We make it a point to update it as new information comes through our research programs. Also, I wanted to thank the North Central Soybean Research Program and the United Soybean Board for funding several of the Diaporthe Phomopsis projects that we have done from my program, as well as in collaboration with others across the country. Thank you.